Okay, so today we are on our lecture number 7, which is going to be Introduction to PCR or Polymerase Chain Reaction. But just to remind you, before we start that next week we don't have a lecture because you guys have your second MCQ. So you're going to have, this time you will have 15 questions and you will have 45 minutes to answer them, and they will be based on lectures 5, 6, and 7. And just like last time, it will open at 12 p.m. and it will close at midnight. So it's going to be next Wednesday, 20th of March. And just like last MCQ, it's going to be worth 10% of your overall grade. And again, negative marking will apply. So minus 10% for incorrect answers, just to make it a little bit more challenging because it is an open book. So you can use whatever you want while you're doing this exam. Okay, and with this now out of the way, we can move to our learning objectives for this week. So we will talk about theory and practice of both qualitative and quantitative PCR, so more like traditional or older PCR, and then qPCR that is now used more, and then we'll list and discuss biopharmaceutical applications of polymerase chain reaction in biopharma. So we moved on our table, so we are now here, and as you can see, PCR can be used again in most of the stages of your biopharmaceutical life, but it's mostly used in characterization for in-process samples and also for lot release. And mostly it is used to help us detect contaminants, and the contaminants that we want to detect are things like pathogens, so things like mycobacteria, for example, mycoplasma, and also host cell DNA. So we want to check whether we have any contamination with host cell DNA, and it's used a lot for raw material testing, for testing of, again, contamination in our raw materials, as well for testing contaminants such as viruses. So it can be used a lot. It's just basic principles of PCR or polymerase chain reaction. It's basically quite rapid and these days relatively inexpensive and simple way of producing millions of copies of DNA or deoxyribonucleic acid from really small amounts of starting material. So it can really start with minute materials and get millions and billions copies of DNA, so you can actually visualize it or detect it in some way. So you can look at PCR as molecular photocopying of your DNA in a test tube. It is used everywhere, so in medicine we use it for genetic analysis, for diagnostics, for pathogen detection as well. Where it's really popular, it's in forensic science, archaeology, where you can use DNA, even small traces of DNA for human identification, and then in biopharmaceuticals, as I said, it can be used to rapidly detect and quantify the amounts of viruses that we might have, residual host cell DNA, or small bacteria such as mycoplasma that is quite hard to detect in some other ways. So just to remind you, our DNA is it's inside our body as double helix that is packed in our chromosomes, and it contains a phosphate deoxyribose backbone and then nucleotides. And they're making our DNA. There are 4 nucleotides that making DNA: G as guanine, A as adenine, T as thymine, and C as cytosine. So you're probably quite familiar with this. But again, just to remind you, building blocks of DNA and RNA are nucleotides, and nucleotides are actually made up of 3 parts. So first thing that you have is a nitrogen ring structure that's called a nitrogenous base, and this ring structure will be different depending on the nucleotide. So you can see up here that we have pyrimidines and we have purines. So this is the structure of cytosine. This is the ring that we find in thymine in DNA. In RNA, we have slightly different version, which is called uracil, and then this is the structure of adenine and the structure of guanine. So they're different depending on the nucleotide. Then we have a 5-carbon sugar, and you can see this sugar is again different depending on whether you have DNA or RNA. In DNA we have deoxyribose, and then in RNA we have a slightly different version, and this is ribose, so this is where they differ as well. And then we have at least one phosphate group that is attached to our 5-carbon sugar. We often talk about complementarity when we talk about these nucleotides, and it's just the way that they bind to each other and create a double helix. So DNA sequences are usually written in the 5' to 3' direction, and what that means is that we are just talking about the carbons that are involved in bonds between different nucleotides. So you can see we have 1' carbon, or the first carbon, that's the carbon that has this nitrogenous ring attached. Then we have 2nd carbon. We have 3rd carbon that contains hydroxyl group. Then we have 4th carbon here, and then 5th carbon contains this phosphate group. Oops. So when new nucleotides are added to a strand of DNA or RNA, the strand grows at its 3 end. So if we have a thymine here, the strand will grow at the 3 end, where the phosphate that's on carbon 5 of incoming nucleotide, in this case guanine, will attach to the hydroxyl group that's here on the 3' end of the chain. So this is how we can write it then. We can write it in this 5' to 3' direction. So we can say we have thymine, then we have guanine, and then whatever is next. On the other side, because our DNA exists as double helix, we have complementary pairs that bind to the left side, let's say, and thymine and adenine always come together, guanine and cytosine in DNA, and they the other part of the chain grows in opposite direction. So that just means that the 5' end of one strand is actually paired up with the 3' end of each matching strand. So this is the complementarity that we are talking about. When doing PCR, there are a few components that you need to have in order to replicate your DNA. So you need your DNA sample that you want to replicate, or your sample which contains the gene that you want to replicate. You need to have primers, so we'll talk about all of those components in more detail, but you need to have primers to actually bracket the area of the whole DNA that you want to amplify, because you don't want to amplify absolutely everything. Then you need to have those building blocks or nucleotides in order to copy DNA. You need to have an enzyme that will help you add nucleotides to a growing chain of DNA. You need to put all of that in a buffer that will provide a good environment for the reaction to happen, and that all needs to be put into a specific tube that will allow for the reaction to happen. All of those components that go into a thermal cycler that is capable of heating our samples to different temperatures in order to multiply our DNA. So we'll talk about all of these steps in more details. So what are primers? Primers are short pieces of DNA. They usually only have 20 to 30 base pair in length, and unlike our DNA, which is double helix, primers are single-stranded pieces of DNA that are made artificially, so companies make them for us. And they need to be made in a way to choose the fragment that we want to amplify from our test DNA. So they are going to decide what part of the DNA will be amplified. And of course, it's going to be us who is deciding that and making primers. They're made to be complementary to the sequence on the test DNA sample, and they are necessary because if we don't have them, then our DNA polymerase wouldn't know where to start and how to actually copy the DNA. So if we have our double-stranded DNA here, we denature it to split it into 2 single-strand pieces, and from all this DNA we decide that we just want to copy this red part, so this is the region of the gene that we want to amplify. We need to make two primers, so short single-stranded DNAs that will be complementary to each side of the gene that we want to amplify. So we have a forward primer that's complementary to this side and reverse primer that is complementary to the side here. So they are basically bracketing, so they anneal to complementary sequences on the opposite strands of the DNA on either side of the region that we want to amplify. And then what's amplified is what's between them. So only the DNA between the primer sequences is amplified by the polymerase enzyme and the rest of the PCR components. So they're extremely important because they guide what's going to be amplified. Then in the mix we have nucleotides, so they are building blocks of our DNA, so we need to have a mixture of adenine, thymine, guanine, and cytosine, and we need to have enough of them. And they also have 3 phosphate groups attached to them, so they're called deoxynucleoside 3-phosphates or DNTPs. So these phosphate groups give energy to the reaction, so they really help with the energy of the reaction. They are, again, you buy the mix of nucleotides from different manufacturers, and usually needs to be kept frozen, but this mixture is quite sensitive to freeze-thaw cycles. So when working with PCR, you just have to make sure that you don't freeze-thaw them too many times, because you can destroy completely nucleotides and then you won't have any building blocks to build your DNA. Then we need an enzyme that's actually going to be putting those nucleotides to their complementary pairs to copy DNA. And in our body, there is a naturally occurring complex of proteins that do that, and they're called DNA polymerase. So DNA polymerase copies our cell DNA before it divides it too, and that's happening all the time in our body. But the DNA polymerase that we use in PCR reaction actually does not come from the one from our body. It comes from a strain of bacteria that is called Thermus aquaticus that actually lives in thermal pool at Yellowstone National Park and some other areas of the world, and it's called Taq polymerase, so that's the most popular one. And that one is really stable at 95 up to 95 degrees. So the reason why we don't use our DNA polymerase, although it was used at the beginning, so the polymerase that we got from E. coli was used in PCR, but the DNA polymerase that is found in prokaryotic and eukaryotic organisms, it degrades pretty easily. So it's made, if you think about it, it works in our body at around 37 degrees, so it really degrades at high temperatures, and in order to copy DNA outside the body, we need high temperatures to denature double helix and make our DNA single-stranded so we can copy it. So at the beginning of PCRs, scientists were using DNA polymerase from, as I said, E. coli, but they had to put quite a lot of amounts at each step. So now we are using DNA polymerase such as Taq, which are thermostable enzymes. They are found in special kind of organisms that are called thermophiles. They like to live at really high temperatures, and it's really active at high temperatures and widely used, so much more stable than DNA polymerase. Taq polymerase is definitely the most popular one that is used for reactions, but there are other polymerases as well. So there are some of them listed here, so such as Pyrococcus furiosus, Thermus thermophilus, deep vent polymerases, and they slightly differ in their processivity, fidelity, and persistence. So processivity means processivity is the rate at which polymerase enzyme makes copies. So things like Taq polymerase adds around 50 to 60 nucleotides per second, so it's pretty quick. Fidelity means it's how many times our polymerase will make a mistake. So they all make mistakes, not often, but they still do. That happens in our body as well, so we get mutations. So for example, Taq polymerase makes error every 57 millionth time in our template. And then persistence is how stable they are at high temperature, and probably something like deep vent polymerase would be the most stable one, which means that you can put it at 95 degrees and it has like half-life of nearly a full day at 95 degrees, so it's extremely stable. All of these components need to be put in a PCR reagent buffer that will provide optimal pH and salt environment for the reaction. Especially we need to have magnesium in our buffer, because magnesium is cofactor and catalyst for polymerase activity. So polymerase needs magnesium in order to work. And then all this needs to be put in a PCR tube, so a special kind of small tube that has a really thin wall, and this thin wall permits thermal conductivity. So it allows whatever is in the tube to be heated and cooled down rapidly in a thermal cycle. And then everything is put in a certain thermal cycler. So this is a piece of equipment that just enables the reaction to be heated at different temperatures that are critical for successful experiments. So we'll talk about those. So usually this is how thermal cycling works. When you put all of those samples into a thermal cycler, first thing that you do is you heat your reaction to 95, 96 degrees, and this denatures DNA, so causes melting of DNA templates because it disrupts hydrogen bonds, and we get single-stranded DNA molecules. So DNA exists as a double helix, but we want to denature it so we can copy it. So we want to make it in a single strand. After that, the reaction is cooled down to around 55 to 65 degrees to allow for primers to bind to their complementary sequences of single-stranded template DNA, so we know what to copy. And then it's heated to around 72 degrees, and this is the temperature at which Taq polymerase starts working, so it starts extending primers and synthesizing new strands of DNA. This cycling is usually repeated 30 to 40 times, and each of them lasts maybe a few seconds or a minute, but you're repeating it over and over again because you want to get more and more copies. So that's shown a bit more here. So if we heat and denature our DNA, separated into two single strands, when we cool it down, primers can anneal to the both sides of the gene that we want to copy. Then Taq polymerase comes in and copies this part of the gene. So that's first cycle. In the first cycle, we get 4 copies or 2 DNA double helices. But then we repeat the reaction, and in the second cycle we get 8 copies, in the third cycle 16 copies, fourth cycle 32 copies, and so on and so forth. So if we were to just kind of repeat this for 30 cycles, we actually end up with 2 billion copies of our DNA. So this is exponential amplification. So we get quite a lot of DNA from small starting material, and now that DNA can be detected in some way because it's not as small anymore. We have quite a large amount. So typically PCR run is 30 to 40 cycles. You don't really go over 40 cycles because DNA duplication at certain point reaches a plateau, because the production of this new DNA, which we call amplicons, so new products, is self-limiting process. So you can see as you start cycling, the first cycles are going to be exponential duplication, when we get duplication in every cycle. But after a while, because we are kind of reusing all the material and we have quite a lot of amplicon present there, we start going into linear phase where we don't produce, we don't duplicate every cycle, and then we reach plateau phase at around 40. So we are not really getting anything anymore. So typically you would stop at 40 cycles. Traditional PCR, so the older PCR that we did, once we finished with all this cycling for 30 or 40 cycles, in order to visualize our DNA, we had to put it onto an agarose gel and do agarose gel electrophoresis. You can do electrophoresis because nucleic acids are inherently negatively charged because of the sugar phosphate backbone, so you don't really have to do anything. You can just put it almost like your SDS page for proteins, but you don't even have to add SDS to make them negatively charged. DNA inherently is already negatively charged. So you put it onto the gel. All the DNA will move towards the anode, but longer strands will be slower, just like again in gel electrophoresis. Longer strands have greater difficulties moving through pores, while smaller DNA strands can move much quicker. So you can separate DNA based on size on an agarose gel. Agarose that is used is polysaccharide. It's just a brown sugar from red algae, and it needs to be dissolved in an electrophoresis buffer and then melted in the microwave in order to get a gel. So we melt it in the microwave, we pour it into a gel mold, and then relieve it at room temperature for, let's say, 20 minutes to solidify. So first you have to heat it or melt it in the microwave to get the formation of this initial gel, and then when you leave that gel aging, so leave it standing at room temperature, you get the final gel structure. And your gel will have different pore sizes depending on the amount of agarose. So the more agarose you add, the smaller pore sizes. So usually you would go with something like 1% or 2% of agarose in a buffer, but it can be different depending on what you are doing. So the way agarose gel electrophoresis would work, again, quite similar to your protein electrophoresis. If you have your different samples, you would also use molecular weight ladders. So remember, in gel electrophoresis, we were using a ladder that you buy from the manufacturer that has a mixture of proteins of known sizes, but in kilodaltons. Here you would have commercially available mixture of nucleic acids of known molecular weight, and the weight would be in in base pairs rather than kilodaltons. And it can be used to estimate molecular weight of samples. So all of your samples and molecular weight ladder are added in the little wells of the gel, and then you run the gel, and samples will travel to the gel depending on their size. So you can have a look at them. Same as proteins, you can't visualize DNA without using a dye. So in protein analysis, we had to run our gel electrophoresis and then stain our proteins with a dye. For DNA, we add nucleic acid stain into the gel mixture before it sets. And so as our DNA is moving through the gel, it actually gets stain, binds to DNA and RNA and causes them to fluoresce under the UV light. Typically, ethidium bromide was used, but ethidium bromide is potentially carcinogenic. So now, wherever you can, you try to use alternatives. So alternative dyes have been developed that bind to DNA and RNA specifically. So things like cyber gold or cyber safe dyes that can be bought again if they're commercially available. So usually you use a UV light to then visualize it, or you can use a blue light transilluminator in some of the instrument, and then you get images such as the one here. So with your molecular weight ladder and then your different bands of your samples. Big disadvantage of traditional PCR is that you are looking at the results from the end point of the reaction. So you have to wait for all the 40 cycles, 30 or 40 cycles to be done, and then you're basically looking at it here. But just to remind you, this is the best phase of PCR because we get exact doubling of product at every cycle, and reaction is specific and precise. But then as we go to around 30, we go into the linear portion of the reaction, where reaction is slowing down and reaction components, because the reaction components are being consumed and product is degrading, and then we really reach this plateau phase where the reaction has stopped and no more products are being made. And this is really where we are looking at if we're doing traditional PCR. And the problem with that one is that there is high variability of the amounts of DNA that is being made beyond the exponential phase. So here we see 3 replicates with the same starting quantity of material from the start, and during exponential phase, early exponential phase, you can see that they all have the same amount of DNA. But as we are going through linear phase and plateau phase, they kind of separate. And even though we started with the same amount, it looks here like they are actually contain different amounts of DNA at the end. So the amount of starting material cannot cannot be accurately quantified using traditional PCR. So traditional PCR gives us poor precision, low sensitivity, and low resolution, and it cannot be automated. We only get size-based discrimination. Results are not expressed as number because, again, we are using a dye for staining, which is only semi-quantitative, so same as when we do densitometry on our protein gels. So if we take these measurements at the plateau phase, data does not truly represent the initial amounts. So exponential phase, remember, it's much more precise and sensitive. So can we use exponential phase to actually determine the amount of starting material of your DNA? And the answer is yes. This is where real-time PCR or quantitative PCR or qPCR is coming for. So quantitative PCR or qPCR uses this theoretical quantitative relationship between amount of starting material and amount of PCR product and everything different cycle numbers. So it allows us to monitor the increase in product formation in real time, so as it occurs in the exponential phase. So we can actually look at the doubling of our material at every phase, okay, and not just at the end, and that enables quantitative determination of the starting amounts of DNA in the sample. So what you're seeing here, again, you're seeing 96 replicates. They all have the same amounts of DNA. And again, if you look at this plateau phase, look at the variability between those 96 replicates. They should all have the same amount of PCR product because they all started with the same initial amount, but look at the difference when you look at the y-axis. So if we go back and actually look at them in exponential phase, you can see that they are all much more similar. So instead of collecting data at the end, with qPCR, we collect data throughout the whole process, and we really concentrate on that exponential phase. So in real-time PCR, the amount of DNA is measured after each cycle, and we can do that because we add fluorescence dye to our DNA as it's being made, and that yields increasing fluorescence signal. And that's proportional to the number of PCR product molecules generated. So the more product we have, the higher the intensity of the fluorescent signal. And for this, we need to use a special instrument. So we are again using a thermal cycler that can do, cycling of all the temperatures. But this instrument also needs to have fluorescent dye scanning capability. So it has to be able to scan across our little tubes or wells and scan the amount of fluorescence that is coming from each sample. And then we can plot fluorescence against cycle number, so cycle number 1 to 40. And our PCR instrument generates an amplification plot that represents the accumulation of product over PCR reaction. So amplification plot looks like this. So amplification plot or amplification curve. What we are seeing here on the x-axis is cycle number, so from 0 to 40. On the y-axis we see relative fluorescence. And then we have all those different curves that corresponds to different starting amount of material. So you can see red one, we are starting with the highest amount of DNA, 10 to the power of 8. And then yellow one is the lowest amount of starting material. And then the gray one is no template, so we actually did not put anything in this one to help us determine, whether our reaction is happening properly. So baseline fluorescence that we have here, so the black line. So baseline of the real-time PCR refers to the signal level during the initial cycles of PCR. So those are usually cycles 3 to 15, where you don't really see fluorescence signal increasing because it's so low because you don't really have enough product yet. So you only made 32 copies or, only hundreds of copies. So you still don't have enough signal to be able to see it. It's just masked by the background of the reaction. You don't know what's the background, what's the noise. What's the noise and what's your signal. So that's your baseline. Then we have a fluorescence threshold. So threshold is the level of signal that reflects a statistically significant increase over the calculated baseline signal. So it's basically telling us, so after this amount of fluorescence, we are going to be able to distinguish between the signal and between the noise. And then we also have the threshold cycle, or CT. CT is the cycle number at which fluorescence signal of the reaction crosses the threshold. So it's basically telling you when is your reaction starting to be positive, when you actually can measure something. So you can see for this red sample, because we started with the highest amount of starting material, it didn't take long to multiply that much to be able to see fluorescence. So after cycle 14, we are able to measure fluorescence, and then we measure fluorescence through all the cycles. And we see that exponential phase, and the linear phase, and plateau phase. For the blue one, for example, we can see that we are crossing this fluorescent threshold, and we have our CT number at around 17, cycle 17, and so on and so forth. So CT is the point at which the reaction returns a positive result, and this is then used to calculate the initial DNA copy numbers, because CT values is inversely related to starting amount of target. So basically, the more starting amount you have, you will reach this CT earlier. Okay, so if you have a sample with twice the amount of starting material, this will yield a CT one cycle earlier than a sample that contains half as many copies of target prior to amplification. Okay, so the earlier the CT just means that you started with the higher amount of starting material. So you can quantify everything at the end. So you can do absolute quantification and relative quantification. For absolute quantification, you would use samples of known quantity, you serially dilute them, and then you amplify them to generate standard curve. So just like any standard curve that we talked about in the last few weeks. And then you can quantify the amount of unknown samples by comparison with this curve. Or you can do relative quantification, where you are looking at the expression of gene of interest in one sample compared to expression of the same gene in another sample. And then you express results as fold change. So for example, you can look at healthy volunteers and volunteers that have some gene mutation, and then you're looking at the, for example, increase of gene compared to control volunteers, and you can say as twofold increase or twofold decrease. So you need to compare it. You're always comparing it to some kind of control in relative quantification. So I said that we can monitor the amount of DNA that is being produced in each cycle because we are measuring fluorescence. And there are a few different chemistries that we can use to monitor the amplification of the target sequence. I'm going to mention the most popular ones, but there is definitely more than these. So the ones that are used the most would be dye-labeled sequence-specific oligonucleotide probes, such as Tachman assays, or DNA binding dyes, like cybergreen. So we'll talk about them. So what Tachman is? So Tachman probe is sequence-specific probe that has a fluorescence on it and that anneals between the forward and reverse primer on the DNA. So it's made to bind to your DNA sequence of interest between your forward and reverse primer. It has dual label, so it has reporter dye and it has a quencher dye on it. And it's designed to have slightly higher annealing temperature compared to PCR primers. So the probe binds a little bit before then primers bind and the extension of primers begin. So basically it looks like this. So you have your little probe that is designed to bind complementary to the part of gene that you are trying to amplify. So in between your reverse primer and forward primer. This probe has a fluorophore that fluoresces and a quencher. So when probe is intact, this fluorophore and quencher are close to each other, and quencher is quenching the fluorescence of the fluorophore. So fluorophore doesn't have any fluorescence. When reaction starts and DNA polymerase starts amplifying the region in between primers, once it comes to the probe, it actually cleaves the probe. And now when it cleaves the probe, this fluorescent tag and the quencher, they are separated, and now this dye, reporter dye, can actually fluoresce because quencher is not there to stop the fluorescence. Okay, so the more DNA we have being made, the more of this Tachman probe will be cleaved, and the more free reporter dye will be in our mixture, so the higher the fluorescence intensity. So again, just to remind you, so first we denature our DNA, then your probe will anneal first. Okay, so probe will anneal first because it's made to anneal before primers. Then the primers will anneal, and then DNA polymerase will start copying the area between primers. And while copying that, when it comes to probe, it will cleave the probe. Reporter dye will be separated from a quencher, and it will start fluorescing. Okay, so that's one chemistry. Next chemistry is cyber green chemistry. So cyber green is a dye that binds to any double-stranded DNA. So wherever you have double-stranded DNA, whatever kind of double-stranded DNA you have, doesn't have any fluorescence when it's free in solution, but then when it binds to DNA, this fluorescence decreases 1,000-fold, so it can be seen. Okay, so the more double-stranded applicons or products are produced, cyber green dye will increase. So again, it's proportional to the amount that we are making. So again, this dye doesn't have fluorescence really, but when it binds to DNA, it starts fluorescing. So cyber green is simple to design. So for doing reaction with cyber green, you just need your two primers, and then you can use cyber green for any kind of primers complexes that you have, for any kind of genes that you're trying to amplify. So you can test multiple genes quickly without designing multiple probes. So remember, probes need to be designed to complementary bind to your gene of interest between your primers. While cyber green binds to any double-stranded DNA, so it doesn't have to be designed separately. You can just buy, like universal cyber green. And you have lower initial cost because probes cost a little bit more than cyber green. But there are quite a lot of disadvantages of cyber green. So the main disadvantage is because it's not specific. So it's not just going to bind to your DNA, it will bind to any double-stranded DNA, any double-strand that you have there. So it cannot distinguish between amplification of your target DNA and then amplification of, for example, primer dimers. So primers, they tend to dimerize themselves, and then they become, so they kind of bind to each other, and then they become double-stranded DNA, and cyber green binds to them. So then it looks like you have more DNA than you have target DNA. Or if you are also amplifying something that's non-target sequence, this will also show on cyber green fluorescence curve, and you won't be able to distinguish between amplification of target sequence or something that's non-target, something that's off-target effect. So again, you will have false positive results. So for this reason, when using cyber green, you need to verify the target DNA that is being amplified. So this is commonly done by using melt curve analysis. So what that means is, like, once you go through all of your 40 cycles, then you heat your product again, and you are measuring melting temperature. So you measure when is your product starting to melt. Okay, so melting temperature is temperature at which DNA is half double and half single stranded. So in this case, when it's half double, half single stranded, cyber green dye fluorescence will decrease. Remember, it's only binding to double stranded. So if you are heating the product and you are making it single stranded, cyber green fluorescence decreases. And then you can plot this melting curve either as fluorescence versus temperature, so you get this melt curve. But what you actually do is we do first derivative of change in fluorescence over change in temperature, and we get melting peaks. So this is much nicer to kind of visually see. So melting peaks, you can see Tm, so this is the temperature at which you melted half of your DNA in this melt curve analysis. And this helps us to see whether we are amplifying something else, because if you're only amplifying your target DNA, you should only have one melting peak. But if you were replicating or multiplying several products, then you will have several peaks. So if you see several peaks, that means that you have off-target effects, or you are seeing things like primer dimers. So for example, here you can see that these samples, they all have just one peak, but there is another one here in the red sample, and it has lower melting temperature. So, and really broad peak, so this usually represents primer dimer. So if you see something like this during your melting curve analysis, you see that you have something that you don't really want there. So PCR is really sensitive technique. So remember, like, you're multiplying DNA from really minute amounts. So if you have any contamination, that will be a problem. So if you have any contamination with something that you don't want to amplify, that gets amplified, that will give you false results. Or you can have contamination with things that can destroy your DNA. So with each PCR study, you have to have multiple assay controls to confirm optimal performance of the assay. You have to have special reagents. You have to confirm absence of reagent contamination and PCR inhibitors. And you need to do it in a separate area. So you want to do it in kind of area that is designated just for doing PCR reactions to stop any contamination from happening. So where do we use PCR in biopharma? In biopharma, mostly we use it for viral detection and quantitation. So viruses are really small, it's so hard to detect them and quantify them. So you want to use DNA to do that, PCR, sorry, to do that. For mycoplasma detection and quantitation, so again, mycoplasma is a really small bacteria that doesn't have a cell wall, so it's hard to quantify and detect that one. For whole cell DNA detection and quantification, because we don't want whole cell DNA in our samples, and it can be used for pathogen detection as well, not so much in biopharma. So mostly in biopharma to detect, as I said, whole cell DNA, and we want to check for them in our cell banks, in our bulk harvest material, and in our finished product. And same thing for viruses. We want to check that our cell banks don't contain viruses, that our bulk harvest material doesn't contain viruses, or our finished product doesn't contain DNA or RNA viruses. Why we worry about whole cell DNA? So we are using, when you're making monoclonal antibodies or biopharmaceuticals, so they're made out of genetically modified host cells, bacterial cells or mammalian cells. So there is going to be some DNA from those cells. And the worry is that if that is in our final product, that can be a safety issue, because if there is any residual host cell DNA present there in sufficient amount, that could transmit genetic information to patients. It hasn't happened yet, but we worry that this might happen. So we want to make sure that we are removing all of that host cell DNA. To do that, so your regulatory authorities tell you that you have to do it, and they give you the amounts that are kind of acceptable. But this can be, again, slightly different on a case-to-case basis. But just for an example, so World Health Organization and European Union tells you that you can have 10 nanograms of residual DNA per dose. FDA is saying that you can have 100 picograms per dose, and you can use some of these kits that are available from different vendors, so like Tachman residual DNA Chinese hamster ovary kit if you're using CHO cells, or residual DNA E. coli kit, but mostly companies need to design kind of their own kits. And then detection of viruses, so lots of our cell lines contain endogenous viruses in them, so we have to make sure that we quantify the amount of these viruses. So they just live in the cell, like just viruses, like some viruses live in our cells, but they don't cause any issues, but we're just worried that they might cause issues if they are injected into a patient. So we need to quantify the amount of viral particles in cell lines, and then we want to check whether we remove those particles during purification. So using our purification strategies, we want to make sure that we don't have that we have low amounts of those particles, viral particles left. And then you can also use it to detect bioburden in raw materials. So for different raw materials, to detect the amount of, something like a bacteria or a virus, again, because you don't want it there. So what this one is showing is just like your traditional PCR on a gel, and testing of different raw materials for E. coli genes. So you can see that they are present there. So you just want to test your raw material, make sure that something like this is not present, for example, or its present is in really low amounts. Okay, so just to summarize, we talked about principles of PCR, both traditional and qPCR or real-time PCR, and applications in biopharma. Okay. Do you have any questions for anything today? No? Okay. No problem, Anda. If you missed anything, this is going to be recorded, so you can listen to it in your own time. Any questions? Okay. MCQ, how do we know our results? The results be sent personally. Can you also see the correct? So usually after the MCQ,